Okay, so next I will have Mark Sherdak, Lance Taylor, Mark Meadle, and Larry Vernetti's team looking at a liver acinus microphysiological system for determining drug candidate dosing in clinical trials of liver disease. Thank you.
Well, thank you, Pasley, for the introduction, and thank you for allowing us to come and tell you about what we're doing with our liver Acinus MPS model. So today what I wanted to do is describe some of the lessons that we've learned while putting together our qualification plan for our liver Acinus MPS system for determining drug candidate dosing in clinical trials with patients with liver disease. And we have just submitted this QP 2 weeks ago. So just a little background, what the unmet need was for our team was to understand how to effectively translate the pharmacokinetic data in healthy volunteers that's generated in the phase 1 to a patient population with liver disease that the drugs may be affecting in phase 2 to understand how to make any recommendations for any dose adjustments in this patient population. Because as you know, the liver is a major organ of metabolism. It also will help determine what the blood plasma levels are. And patients with hepatic impairment or liver disease can have impaired metabolism, which can potentially lead to increased and potentially toxic levels in the blood. And so our goal with this particular aim was to qualify our biomimetic MPS as a drug development tool to address this unmet need with the specific context of use of the LAMPs as a DDT to establish the hepatic clearance in drug candidates in patients with MASLD, which is probably the most common chronic liver disease today, and assist in the determination of drug candidate dosing in clinical trials when these patients are included. So an overview of the model that we're qualifying. This is our liver Acinus MPS model. It is a 3D layered biomimetic model constructed with 4 liver cell types. It mimics the physiology of the liver Acinus. And this model is amenable to inducing disease states. And so we can induce a metabolic dysfunction-associated steatotic liver disease, or MASLD, from phases F0 to F3. Now, this model is applicable to studying ADME, studying tox, studying liver disease progression, and for precision medicine when patient iPSCs derived liver cells are used. And for the ADME tox studies, it's useful for comparing between a non-disease state and a disease state. Now, the 4 hepatic cell types are necessary for this model to induce the disease. Obviously, the hepatocytes are there for metabolism. They also accumulate fat during the disease. The liver sinusoidal endothelial cells contribute to the pro-inflammatory response of the disease. Hepatic stellate cells contribute to the fibrotic response of the disease. And the Kupffer-like cells in the model are macrophage immune systems to initiate the immune response. Now, the inclusion of these 4 cell types gives this model several advantages over simpler models of just primary hepatocytes or microsomes. One, we can induce a disease state, which is typically not capable in the primary hepatocytes or microsomes. We can look at stability of compounds over a longer period of time in terms of days instead of hours. We can do exposure in a steady state as well as a non-steady state. And we can also induce different metabolic zonation to look at the effect of the disease in zone 1 versus zone 3, for example. So when writing up our qualification plan, some of the lessons learned regarding our model was to explain the role of all of the cell types in the LAMPs. What are they doing? Why are they there? And to justify why this model is better than the more simplified models, as well as to define what the limitation of the lamps is. In this case, we're limited to, say, F0 through F3 of the MASLD disease stages. Now, the LAMS is to be used as a co-decisional tool to recommend dose adjustment in MASLD patients. And this is after phase 1, when we know what the recommended dose in healthy patients are. Currently, when MASLD patients are going to be included, or other liver injury patients going to be included in the study, if the drug meets a certain set of criteria, a dedicated hepatic impairment study would be run. Now, these studies can be lengthy, they can be expensive, and it can be challenging to get patients in for this study. So our vision was to be able to take our LAMPS model and in parallel with the hepatic impairment studies, assess what dose recommendations might be useful for this patient population. As we develop this in parallel with the hepatic impairment studies, when we get comfortable with the robustness and the reliability, it potentially can be a replacement for the hepatic impairment studies. So the lesson that we learned when putting the QP together here was to clearly describe what the role of the LAMPS would be in the regulatory decision process, and accurately depict it with respect to our proposed context of use. Now, the analytical approach for recommending dose adjustments in MASLD with using our LAMPS model, it's multiple steps. The first thing we have to do is we have to establish the LAMPS performance, put the cells together, establish that the cells are viable, establish that the hepatocytes are functioning, and we have to establish that in the case where we're inducing the disease, we actually have the disease. And once that is established, and we do have a set of reference values, which are the criteria that they have to meet to say, yes, the model is functional, we can then add the drug, and we can measure the disappearance of the drug over time in the model. Typically, we run this out to 8 to 10 days. And then once we measure the drug concentration, both what's going into the model and what's coming out of the model, we can determine what the hepatic extraction ratio is, i.e. how much is being metabolized, how much is being lost due to metabolism by the hepatocytes. With that information, we can then predict the hepatic clearance, both in the disease state as well as in the non-disease state. Then we can simulate the drug plasma concentration, saying, well, here's what we're predicting. If you were to go with this same dose in both the healthy and disease state, this is how much more is going to be in the disease state. And then we can calculate what the dose adjustment might be, and this would be a recommendation of perhaps you want to reduce the dose by X amount to get from the disease levels back down to the non-disease levels. And so in writing the QP, some of the lessons learned is to be comprehensive in describing the analytical approach, the entire workflow here. Provide the methods for the calculations, provide the analytical reference criteria that we're going to use, as well as provide data for every step along the way in the workflow. Now, when we initially proposed this in our LOI, we proposed 8 drugs to use for validation, 3 of which in the literature had no dose adjustment recommendations, and 4 which had different levels of dose adjustment recommendations. In our LOI, when it was accepted, the FDA said, well, you might want to expand that number of drugs, and you might want to justify why you're using those particular drugs. And so we've added a number of drugs to the list. And we've also added, okay, here is in the literature what the fold increase in plasma levels are in the different disease states of a hepatic impairment relative to the non-disease state. And so we have a more complete list here with a range of different plasma change levels in the disease. And so, the lessons that we learned here when writing the QP is to justify the number and selection of the drugs for the validation, include drugs with a wide range of clinical dose adjustment needs, as well as provide reference quantitative clinical data to back up those validations. Now, a very important thing is that we're dealing with an in vitro model here, and we need to identify what patients are going to benefit from this drug development tool. And so we need to calibrate our LAMPS phenotypes with the clinical disease stage in order to identify this patient population. Now, in MASLD, there are a couple of ways that they stage the disease in the clinic. They have a fibrosis score, F0 through F4, which is basically determined by the degree of fibrosis that they see in liver biopsies. They also have the Nash-CERN NAFLD activity score, which is based primarily on looking at histopathological samples, looking at steatosis, lobular inflammation, and hepatocyte ballooning. Our model, we can measure steatosis, and we do measure steatosis. We can look at hepatocyte ballooning, and we can assess inflammation using the expression or release of different cytokines. And so what we're doing is we are calibrating our metrics with metrics from a series of histopathology slides from patients from F0 to F4, so that we can say under these disease conditions that we're inducing, they match an F1 or they match an F2 or 3 patient. And so these would be the patients that will most benefit from the predictions from this tool. And so the lesson learned in doing all of this is that we need to keep the clinical context in mind, and we have to link the in vitro metrics that we're getting to the clinical metrics established for concordance. Now, there are a number of other lessons that we've learned as we're writing up to QP. Writing up to QP is very educational because it makes you think about things you didn't think about. It makes you research things that you never thought you were going to research, but it was fun. But one of the, a few of the things is keep up with the FDA updates on their website. The FDA has a lot of information on their websites. They update them every once in a while. Keep up to date with the latest. Take a learn and a confirm approach when establishing the clinical concordance platform. Start off small with some drugs to say, okay, this is what the platform can do, and then confirm it with a larger drug set. We've learned that recommendations of the drug development tool can be categorical and not necessarily specific numbers. Break it down. These are, this range of drugs don't have to be adjusted. This may be a 2 or 3-fold adjustment. Maybe in these other drugs, don't even try it in these patients. It's going to be too high of a drug level. We don't need to say you have to drop this by 2.65-fold. That's not what we want to do. And in doing all of this, we realized that the number of studies required and the amount of data being analyzed is much greater than we originally anticipated when writing up this whole this whole thing, which is presents some challenges, but obviously it's necessary. And so that makes data management even more important. And using a standardized database makes the analysis a lot easier rather than sending out spreadsheets all over the place. And we do put all of our data into the Eve Analytics database for this project. The SOPs are important. We do include SOPs for every step along the way, including how we're doing the analysis for albumin or LDH, for example. Very important to have those. But we also need to strive for GLP-like performance in order to maintain consistency. And finally, you need to know the specific characteristics of your drugs, your samples, and your analytes. We learned early on, imiprazole is not stable at 37 degrees. And once we understood that, we can make adjustments in how we're doing the analysis, and things started to work out. Similarly, we learned LDH activity is not stable in long-term storage. Once we knew that, we want to prioritize analyzing those samples earlier rather than later to minimize any variability just due to that aspect of it. So these are some of the lessons that we've learned while putting together our qualification plan. And the question is, how well have we learned this lesson? Well, in order to answer that question, we look forward to feedback from ISTAND. We just submitted this just 2 weeks ago, so we're anxiously looking forward for that information to go through and make this a better qualification plan. And so then I just wanted to acknowledge the group that is responsible and involved in this project since the beginning.